Hello students, good evening. Welcome back to a new video of Sunanda's tutorial. So in this video we will discuss the important points of the chapter growth and development of plants. It is specifically important video for SSB TGT aspirants, those who have their exams in June. So without wasting any time, let's start. Here in this video I will discuss important points, that means the mind map of the chapter plant growth and development. So at first let us discuss what is growth and what is development. So growth actually is a quantitative term and can be calculated quantitatively, but development is quantitative as well as qualitative term. Growth is fundamental and conspicuous characteristics of a living organism. It is irreversible increase in dry weight, size, mass or volume of a cell, organ or overall organism. Development consists of growth and differentiation, and development is the sequence of all events that an organism undergoes during its life cycle. So during the growth, meristematic cells are totipotent cells which have the capability to divide and redivide. They undergo differentiation, that means a specific role is provided to meristematic cell and they become permanent cell by the process of differentiation. Then permanent cell again acquire and again behave as meristematic cell in the process of de-differentiation. This meristematic cell again provided a specific duty by the process of redifferentiation. So what is meristematic cell? These are undifferentiated totipotent cells which have the capacity of division and redivision. Then differentiation, it is the process of gene directed specialization of a meristematic cell or tissues or organ to perform a specific function. Then de-differentiation, sometimes those permanent cells which have occupied a specific role again regain the power of division and become meristematic through the process of de-differentiation. Then what is redifferentiation? When these de-differentiated cells lose the power of division and become mature again to perform a specific function by the process of redifferentiation. Okay. Next, development. The definition of development: sequence of all events, all changes that an organism undergoes during its life cycle from germination of seed to senescence and ultimately death. So what is seed germination, which is the first step of plant growth? It involves sprouting of seed and growth of embryonal axis. Germination process can be divided into two phases. At first, preparatory phase or biochemical change, which prepares the seed to undergo germination. Then the actual embryo emergence or seed germination. In favorable environment, seed releases radicle and plumule. Radicle which develop to form root and plumule which develop to form shoot. Cotyledon is the first tender leaf originates or emerges when seed germinates. The part above the cotyledon, the embryonal axis part of the embryonal axis above the cotyledon is called epicotyle, and the portion below the cotyledon called hypocotyle. Hypocotyle terminates with the radicle and epicotyle terminates with the plumule. Then the types of germination: one is hypogeal germination, second one is epigeal germination, third one is vivipary germination. What is hypogeal germination? Hypo means below. So cotyledons, that means the first tender leaf remains inside the soil due to rapid elongation of epicotyle. In this germination, at starting phase, cotyledons remains inside soil due to rapid elongation of epicotyle. Example of this hypogeal germination or the plants which shows the hypogeal germination are all monocots, gram, pea, groundnut, etc. Then epigeal germination, cotyledons comes out of the soil due to rapid elongation of hypocotyle. Example which shows epigeal germination, castor, coconut, papaya, etc. Then vivipary germination, germination of seed while it is still present inside the fruit and fruit is attached to the parent plant. The example of vivipary germination is mangrove plants. Then plant growth regulators. What is plant growth regulators? These are organic compounds which influence or controls the physiological activities of the plant. These are divided broadly into two groups. One is plant growth promoter, which enhances the plant growth, and another one is plant growth inhibitor, which inhibits the growth of the plant. Plant growth promoter includes auxin, gibberellin and cytokinin. First one is auxin. F.W. Wendt in 1928 first isolated auxin, and Charles Darwin first studied and proved it experimentally, the role of auxin experimentally, and the precursor of this auxin is tryptophan. From tryptophan auxin is synthesized. Then what are the roles of this auxin? Then physiological roles of the auxin. First, it stimulates cell elongation. It controls the cell division in vascular cambium from where the secondary growth starts. Then stimulate xylem differentiation. It prevents the premature abscission of leaves, flower and fruits. It stimulates the apical dominance. What is apical dominance? The apical part of the plant or shoot apical meristem dominates because high concentration of auxin is present at the apical part of the plant. So it dominates the overall growth of the lateral buds or lateral branches. So that is called apical dominance. The apical part dominates the all other part of the plant. So auxins stimulate or induce the apical dominance. Then it promotes growth of the stem and inhibit growth of the root. Then it causes bending of coleoptile and control the geotropism. Then gibberellin, first isolated from a fungus Gibberella fusariae that causes bacchane or fully seedling disease of rice. Then what are the physiological roles of gibberellin? First one is cell elongation. Then it helps in seed germination. It causes the increase in the length of internode. External application or exogenous application can break the dormancy of seed and bud. It promotes it promotes flowering in long day plant. Promote formation of pistillate flowers in maize. What is pistillate flower? Female flower. And staminate flower in spinach, cucumber and hemp. What is staminate flower? Male flower. Develop antheridia on adjacent gametophyte. In case of bryophytes, it induces to develop antheridia on their adjacent gametophyte. So these are called as pheromones. Then cytokinin, it stimulates cell division, enlargement of stem cells rather than longitudinal extension, help to overcome apical dominance. It promotes the formation of new leaves and chloroplast. It increases number of buds in funaria, regulate morphogenesis, then stimulate release of dormancy in seed and bud, induce flowering in your short day plants. High concentration of cytokinin also induces epinasty, that means curvature of leaves, downward curvature of leaves. Also, it increases the resistance of the plant to adverse condition. Then two plant growth inhibitors. One is ethylene, which is one and only one gaseous hormones. It promotes fruit ripening and senescence, accelerate abscission of plant organ like flowers and fruits, break seed dormancy of cereals, break bud dormancy of potato tubers, induces adventitious rooting and root hair formation, induces leaf epinasty. What is epinasty? Downward curvature of leaf. Commercially used ethylene is ethiphone. Then abscisic acid, which is otherwise known as stress hormone, promotes bud and seed dormancy, promote abscission of leaf and flower as well as fruit. It causes leaf senescence, that means premature falling. Then abscisic acid induces stomatal closure, increases tolerance of plants to various kinds of stresses. Abscisic acid acts as antagonist to gibberellic acid, so it is called as anti-gibberellin. Then what is seed dormancy? Dormancy refers to the condition of seed when it is unable to germinate due to some endogenous factor, sometimes external factor, during which the metabolic activities of the seed is greatly reduced. What are the natural ways to overcome seed dormancy? One is weakening of hard seed coat by microbial action, rupturing or weakening of seed coat by mechanical abrasion, action of digestive enzymes which are present in the alimentary canals of birds and other animals which happen to feed on their fruits, leaching of inhibitors present on the seed coat naturally, completion of over ripening period, inactivation or oxidation of inhibitors by heat or cold or light treatment, production of growth hormones which can counteract the effect of inhibitors, attainment of maturity of embryos in case of dormancy is due to incomplete development of embryos. Then what are the artificial ways? At first, scarification. Scarification means by applying mechanical pressure, we can rupture the seed coat and break the seed dormancy. Then applying hydraulic pressure, treatment with hot water. Then stratification means subjecting moist seed in presence of oxygen to a period of low or high temperature. Then treatment with concentrated sulfuric acid. We can also apply or soak seed in potassium nitrate, ethylene chlorohydrin, thiourea, gibberellin. So these are the possible artificial ways to overcome seed dormancy. Then what is photoperiodism? Physiological response of a plant to the length of photoperiod or light period. The response of a plant to the relative length of light as well as dark period is called photoperiodism. Garner and Allard in 1920 first established the phenomenon of photoperiodism in Maryland mammoth plant, which is a variety of tobacco. But what is critical day length or critical photoperiod? It is a specific day length of light which is required to initiate flowering, or a minimum day length which is required to induce flowering in any plant. So according to the need of light period of a plant, they are categorized into three categories. One is short day plant, second one is long day plant, third one is day neutral plant. Short day plant which require a photoperiod shorter than critical photoperiod to induce flowering. These are also called long night plant. Examples: soybean, sugarcane, dahlia, chrysanthemum, rice, tobacco and xanthium, etc. Then long day plant which require a photoperiod longer than the critical photoperiod to induce flowering. For example, sugar beet, spinach, radish, plantago, wheat, barley, clover, lettuce, etc. Then what is day neutral plant? Day neutral plants actually do not depend on any photoperiod to induce flowering. They are independent of such photoperiod. And the example of it is cucumber, cotton, potato, maize, sunflower, garden pea, maize and chili, etc. Next is vernalization or the chilling treatment or the low temperature treatment to a plant for inducing early flowering. Vernalization stimulus is perceived by embryo of the seed and by the shoot apex in the matured plant. So we can also apply gibberellin instead of vernalization treatment to induce flowering. Then what is phototropism? What is phototropism? It is the movement of shoot towards the light. Shoot apical meristem is positively phototropic. That means they move towards light. But the root is negatively phototropic. That means they grow towards or away the light. Then geotropism, it is the movement of anything towards the gravity. Root grows towards the gravity, so that is positively geotropic, and shoot grows away from the gravity, so that is called as negatively geotropic. So it is all about your plant growth and development chapter. I hope you will get benefit from these important points. So if you like this video, please like it, share with your other friends which are those who are preparing for this type of competitive examination, especially SSB, TGT, PGT and lectureship examination. And please subscribe to my channel Sunanda's Tutorial to get more videos like this. Thank you.